Hey, so if you've got a biology quiz coming up, you are in exactly the right place. Today, we're going to break down enzymes, the tiny little powerhouses that literally make life possible.
So let me ask you this. Is it weird to have a favorite protein? I'm going to say no, because my favorite protein actually reminds me of one of my all-time favorite childhood games. And, believe it or not, this little connection is going to be the key to unlocking how all of this stuff works.
All right, let's get into it. Think of this as your personal study guide. We're going to start with what an enzyme even is, then get into why its shape is so incredibly important. After that, we'll cover how they work, look at some examples inside your own body, and then finally, what happens when they just stop working. Let's do this. First things first, what exactly is an enzyme? To make this idea really stick, we're going to use an analogy you won't forget. Think of them as the Pac-Man of proteins.
Just think about the game for a second. Pac-Man's whole mission is to gobble up those little pebbles that fit perfectly into its mouth, right? Well, enzymes, which are just a type of protein, they work in a really similar way. Each one has a very specific job to do.
Okay, here's the official definition you're going to want for that quiz. An enzyme is a biological catalyst. And that word catalyst, that's the key. It just means its job is to speed up chemical reactions. The reactions our bodies need to survive, which would happen way too slowly on their own.
And here's the most important part about them being catalysts. An enzyme doesn't get used up in the process. Just like Pac-Man can eat pebble after pebble, one single enzyme can do its job on molecule after molecule over and over and over again. It makes them unbelievably efficient.
So now we've got to talk about what is, without a doubt, the most important concept for this whole topic: shape. For a protein, especially an enzyme, everything, and I mean everything, comes down to its very specific 3D structure. It really is all about the fold.
This quote right here just says it all. Having the right shape is everything for a protein to do its job. If the shape is wrong, its function is gone. It's truly that simple. So the big question is, how does a protein get its shape?
Well, a protein's final shape is built in 4 stages. First, you have the primary structure, which is just the unique chain of amino acids your DNA codes for. Then, that chain starts to coil and fold into little shapes, like helices or sheets. That's the secondary structure. The third level, tertiary, that's when it all folds up into its final, compact 3D shape. And then sometimes, you get a quaternary structure, where a few of these polypeptide chains team up to form one giant protein.
Okay, so we know enzymes are proteins, and they have a very specific shape. Now let's see exactly how that shape lets them work. The best way to think about it is like a perfect, really specific handshake.
The first player in this handshake is called the substrate. This is the one specific molecule that the enzyme is built to work on. Going back to our analogy, the substrate is the little pebble Pac-Man eats. In real life, it could be a sugar or a fat or another protein.
And the second player? That's the active site. This is the special spot on the enzyme that is shaped perfectly to fit the substrate. It's Pac-Man's mouth. And this very specific shape is the reason an enzyme that breaks down sugar can't do anything to a fat molecule. They just don't fit.
Now, pay attention to this slide because this is a classic quiz question. There are 2 main models scientists used to describe this whole interaction. The old lock and key model said the 2 pieces fit together perfectly, like a key and a lock, super rigid. But the newer, more accepted, induced fit model says the active site is actually a little bit flexible. It changes its shape just slightly to get a perfect grip on the substrate. And I love this description of the induced fit model. It's not a rigid click. It's more like an enzyme-substrate hug. The enzyme actually wraps around the substrate, and that little squeeze can help the chemical reaction happen faster. You should definitely remember that phrase for your test, an enzyme-substrate hug.
All right, enough with the models and theory. Let's look at where these little powerhouses are getting to work right now, inside your own body, specifically in your digestive system.
And hey, here's a quick tip that could totally score you some easy points on your quiz. Just notice the naming pattern. Enzymes usually end in ase, and the sugars they work on, they often end in O's. So the enzyme lactase works on the sugar lactose. See? Simple.
This slide is a great one to screenshot for your notes. Seriously. It lays out some of the major digestive enzymes. You've got amylase for starch, lipase for fats, and protease for proteins. Just notice how each one is a total specialist, perfectly shaped for one job and one job only. It's also worth knowing that enzymes don't always fly solo.
Sometimes they have these little helpers called cofactors and coenzymes. You can think of them like assistants that help the enzyme get a better grip on the substrate, just to make sure the reaction goes off without a hitch. So we've hammered this home. An enzyme's shape is everything. But what happens if that shape gets broken? And that brings us to our last really important section. What makes enzymes stop working? Turns out, enzymes are incredibly picky. They have an ideal temperature and an ideal pH range where they work their best. If you take them out of that comfort zone, they start to fail. In our Pac-Man analogy, think of extreme conditions as being like the ghosts that can stop Pac-Man dead in his tracks.
And this chart is the perfect example. An enzyme in your stomach, like protease, it loves a super acidic environment with a pH of around 2. But an enzyme in your intestine, it needs the total opposite, an alkaline environment, around pH 8. If you were to swap their places, neither one would work at all.
When the environment gets too extreme, too hot, too acidic, too whatever, the enzyme goes through something called denaturation. And this is not just a temporary shutdown. It is a permanent, irreversible change to the protein's 3D shape. The bonds that hold it together literally break. The enzyme unravels, and it's completely useless.
And that leads us to this quote, which is maybe a little dramatic, but it's totally accurate. When an enzyme denatures, its shape gets completely distorted, the active site is gone, it can't grab its substrate anymore, it is, for all intents and purposes, finished.
So we'll end on this final thought. We've seen that enzymes are absolutely essential for life, but they're also pretty fragile. So given that they control countless critical processes in our bodies, just think about this. How might the malfunction of specific enzymes, or just not having them at all, be connected to human diseases? It's a huge area of medical research and definitely something to think about. All right, that's it for this one. Good luck on your quiz.